נתחיל? So, yesterday we used the PCA, but I did mostly stuff that's non-standard. People don't usually do this type of analysis, and the goal yesterday was to give you a strong feeling of what PCA are, the information that they provide, and what is the meaning of these things. It's the problem with these decomposition methods is that usually they are used as black box. So my goal was to, in a way, yesterday, to open the black box a bit. Not in terms of the mathematics that is involved, but in terms of the kind of information that PCA provides. Today, I'm going to do, in a way, an extended exercise that's pretty standard. Okay, this is the way that you do spike sorting. Okay, so I'm going to go step by step through the different procedures that people do and that you may do. Okay, and in a way it will be more technical. I think that's how things are done. Okay, now eventually you will see that the eyeballing works best, but I'll try to give some hints about how to do things a bit more automatically. So first of all, let's look at the data. These are recordings from cerebellum, again from the lab of Yossi Yarom. This is extracellular recordings. I think they were recorded in loose patch, and you can see a number of spike heights. Okay, there are big spikes, intermediate spikes, maybe small spikes. Okay, and at least some of the spikes have this very deep negative deflection. These are not intracellular recordings, so it's not hyperpolarization of the neuron. It's something. Okay, it's an electrical event that expresses itself as this type of deep negativity on the electrode. So this is the data, and now we want to detect the spikes. Okay, so the first thing, when you do spike sorting, you first detect the spike candidates, and then you sort the spike candidates. Okay, so we want to identify things that might be spikes, and as we said yesterday, spikes are relatively easy to identify because they are narrow and large. Okay, so we expect to have, in this case, they would have a single maximum in the data that you will sort, say, tomorrow. Okay, they will have a large minimum because they have been recorded with metal, extracellular metal electrode. So the spikes in this case have a single maximum, which is preceded and followed by very fast drops. So usually you will identify spikes by their peak size and the speed of the change of the potential. Okay, right. In this case, the signal-to-noise ratio is good enough to find spike candidates simply by local maxima. So the question is how to specify, what do I mean by large? Okay, this things boils down to the question of what is exactly large. Okay, so first of all, you can eyeball, but the standard tool for identifying large small values is the histogram. Okay, so let's look at the histogram of all values. So I provide here two different ways of calling histogram. So this is the array, and that's the number of bins. By default, if you don't provide the number of bins, HIST use 10 bins. Here I have so many data points that I can use many more bins to get very high resolution. The rule of thumb here is that the number of bins should be not more than the square root of the number of data points. Okay, here I have many more than 1 million data points, so 1,000 bins is okay. There's no good reason for the square root rule, but that's the rule of thumb. Okay, so where are my spikes? Oh yeah, before I continue, I mean the other syntax for HIST asks, so if you call HIST like that, it draws the histogram. If you call HIST and ask for return variable, it doesn't draw the histogram. It gives you the counts and the bin center, and you can use the function bar in order to draw the histogram. So these two are equivalent. Sometimes you want to have this array of bin centers for various things. For example, for calculating another histogram with the same binization. Okay, so sometimes this is useful. They are exactly the same. Anyway, where are the spikes? Where are they? The right tail. Okay, so why do I have this huge peak? These are the fluctuation on membrane potential between spikes. So most of the time is occupied by non-spikes. Okay, spikes, even in this trace that has a relatively high rate of spikes. Okay, the spikes are only a tiny percentage of all data points. Okay, so I want to look what happened here in the. So I have here, I mean, I get here a kind of a guideline. So I had large spikes, so these are presumably points that come from the large spikes. Okay, I don't know what this is. Okay, and at some point I start to get the data points that belong to the membrane potential fluctuation that are not spikes. Okay, so looking looking at this histogram, I would say that 0.5 here might be kind of a threshold. So the question, suppose you don't want to look at the histogram. Okay, you want to calculate the threshold as automatically as possible without having to look at the histogram. So how do you how do you formalize what we did now by eye, okay, in terms of the, so that you can run it automatically. So basically what we did was, okay, we are looking for things that deviates from a typical value by a lot. Okay, so in this case, okay, the typical value is here, okay, plus minus, okay, and we want the thing that deviates enough from it, okay, 5,000 and more. So what we want to do is to find a typical value, okay, and find a yardstick, okay, for calculating what is a large deviation from it. Okay, all of this should be familiar to you, and usually we use the mean and the standard deviation. The problem is that mean and standard deviation are the right things to do when you use when you have Gaussian variables and things like that. With spikes, okay, the problem is that you have so-called outliers. Okay, there are points that deviates a lot, and that these type of long-tail distribution are precisely the distributions that are not Gaussian. Okay, that's these type of heavy tails are precisely the thing that causes non-Gaussian behavior in. So you learned probably about the median as a kind of an alternative to the mean. So that's another way of identifying a typical value. What other typical values do you know, by the way? The third one that you, the mode, yeah, the mode. Mode is the number that occurs, it would be the peak of the histogram, which may not be either the median or the mean. Okay, so these are typical values, and each one of them can be, under some conditions, make more sense than others. And for the deviations, usually you're not being taught about it, but there's an alternative to the standard deviation that's called the MAD. It's the median of the absolute deviations from the median, okay, from the typical value. So what I want to do for a few minutes is to do one exercise to demonstrate the difference, okay, the difference in the presence of outliers. Okay, so let's start by theory. When I say, usually people will tell you that the mean is better than the median when the data is Gaussian. Okay, so what do they mean that mean is better than median? People have in mind the following exercise. You generate, let's say, 10 random numbers, calculate their mean, and you ask to what extent the mean is close to the expected value. Okay, so I'm running this exercise here. I generate 10 random numbers, okay, and in order to see how close they are to the mean, I'm going to repeat this exercise 100 times. Okay, so I generate here 100 sets of 10 random numbers distributed normally, so like Gaussian with mean 0 and standard deviation of 1. Okay, I'm going to calculate, so here they are. So I have here 100 lines. Okay, and now I'm going to calculate the mean of each one of these lines, okay, 10 values every time. All of these numbers are estimate of 0, okay, because the mean, the expected value here is 0. And the question that I'm going to ask is how close these numbers really are to 0. Okay, and the way that people do it is by calculating the standard deviation of these averages. Okay, this is about the standard error, okay, the standard deviation of the means. Before even I'm calculating these things, if I take the mean of 10 numbers, okay, that have variance 1, I mean, normally distributed, variance standard deviation of 1, and mean of 0, okay, I calculate now the mean of 10 numbers, okay, I have 100 means of 10 numbers, okay, what should their standard deviation be? What? Yeah, okay. So standard deviation decrease like 1 over n, where n is the number of things that we average. Here we average 10 things at a time. Okay, so all of these numbers should be 0 plus minus 1 over square root of 10, which is about 1/3. Okay, so I expect all of these means to be within 1/3 of 0, so plus minus 1/3. Okay, that's one. Now I'm going to do the same thing, except for instead of taking the mean, I'm taking the median. Okay, and I'm also going to look at the standard deviation of all the medians. Okay, now these are medians of 10 numbers. Every time 100. So what you see, the red line, the blue line are the means of 10 numbers, and the red line are the medians of 10 numbers. Okay, so first of all, they are within 1/3 of 0, okay, as we expected. Okay, this is 1/3. Okay, so they lie mostly between 1/3 of 0. Okay, they are not very different from each other. Now, how shall I select between them? Okay, so I'm looking for when people say that the mean is better than median, okay, what they mean is that the standard deviation of the blue number is smaller than the standard deviation of the red numbers. Okay, you see that the red numbers tend to be a bit more extreme. Okay, the blue are, and this is what happens, okay, not by much. This is a standard deviation of the means, and this is a standard deviation of the medians. Okay, so the fact that the standard deviation of the means is smaller than the standard deviation of the medians is the reason why people say that the mean is better than the median. Okay, that's a numerical measure. It's not much better. Okay, I don't remember now the number, but it's something like 14%. I mean, you can calculate the things exactly. And it comes out to be something like 14%. Okay, that's yes.
Would the mass of the mean be smaller than the mass of the median?
The MAD has a slightly different scale. Okay, we'll get to the MAD in a moment. Okay, right now I'm talking about the medians, not about the MAD. Okay, at the moment. I don't know, but you can test it, okay? You can check it. Okay, now I'm going to do repeat the exercise now. So this is for Gaussian numbers, okay? I'm not, can run. No, so each one of the individual numbers at this, okay, at this stage, each one of the individual number has a standard deviation of 1. Okay, so when I take the mean, the standard deviation is going to be 1/3. I'm doing it 100 times and calculating the standard deviation, so I should get something that's close to 1/3. Okay. Yes, we.
I don't understand. You said the mean is better because mean is smaller?
It's closer to 0. It's closer to 0. The standard deviation, the blue line is, I mean, not by much, okay, but the blue line is closer, on average, is closer to 0.
Isn't it more close than 1 over 10
It,
square root of 10? Is that a representation of the derivative value rather than 1/0?
you mean a theoretical representation of 1/3?
Yes.
Well, I'm not trying to, again, these numbers, all of the blue numbers try to represent 0. Okay, so what I want them is to be as close to 0 as possible, okay, and this is what's shown here. Now, these are noisy estimates of 1/3. I don't know, I'm not trying to estimate 1/3. If I run another run, okay, they will be very different. They may come to be larger than 1/3 and so on. So the fact that in this case, this came to be closer to 1/3 than that is by chance. Okay, I can run it again. Okay, here I run again okay, so here, for example, okay. So now, again, this is larger than that. Okay, but now that one is closer to 1/3. Okay? Okay, so let's look at data that's not Gaussian. Okay, and the way to make the data not Gaussian is by introducing by hand an outlier. Okay, so what I'm going to do is, again, to select random numbers. 9 of them are going to be with variance 1, and the 10th, okay, I select to be with variance 10. Variance 100. A standard deviation of 100. Okay? So most of them will be typically 1, and the last one will be, no, excuse me, variance, variance 100, standard deviation of 10. Okay? So most of them are close to 1, okay, and the last one, okay, is typical size 10. Okay, and now I'm going to look, so again, the point is that the average value is still 0. Okay? The average value is still 0 because all these 9s have average value of expected value of 0, and that one also has an average value of 0. Okay, so all the average value of all 10 numbers, the expected value of all 10 numbers is still 0. Okay, it's just that the statistics changed. Okay, the last one may be highly variable. Okay, and now I'm going to repeat the same calculation. So I'm calculating the mean of all numbers and the median of all numbers, and then I'm looking how close the means and the standard deviations are to 0. Blue is the mean, okay, and red is the standard deviations. And red are the medians. Oh. Blue are the means of the 10 numbers, and red are the medians of the 10 numbers. Okay, now the medians are obviously much closer to 0 than the means. Okay, and this is, again, it's reflected here. So the standard deviation of my means is much larger than the standard deviation of the median. The standard deviation of the median is going to be also a bit larger than what it was before, but only a little bit larger, whereas the standard deviation of the means increased by a lot. Okay, so the way people, so in the presence of outliers, okay, the median is much better than the mean. Okay, this is a lesson. The way people express it is by saying that the means is not robust, the median is robust. Robust means that introducing an outlier that may be as large as you want changes the median by a bounded value, bounded amount, whereas the mean will change in an unbounded way. Okay, if you can add a very large number, and the mean will just move with it. Okay, so in the same sense, the standard deviation is not robust and the MAD is robust. Okay, so in the presence of outliers, the mean and the standard deviation may suffer, whereas the median and the MAD stay robust. Okay, so all of that is something to keep in mind, and the question is, what does this mean? Okay, what does this mean to this larger selection in the context of spike sorting? Okay, so there is a sense in which mean uses data better. Certainly, it's optimal for Gaussian signals. Okay, the median is slightly more noisy on Gaussian signals, but performs much better in the presence of outliers. Okay, this is what I showed you. Okay, that's same thing about standard deviation and the median and the MAD. Okay, so STD uses data better and is optimal for Gaussian signals in a sense that can be expressed exactly. And the MAD is a better descriptor of typical deviation in the presence of outliers. Okay, it doesn't suffer so much from the... Now, the rule of thumb for using mean and the robust versus non-robust is that if you have a small samples, mean and standard deviation are better because the optimal use are preferable. I mean, they are the first thing to use. Okay, and the reason is that the lack of efficiency of the median and the MAD may express themselves. That's one. And secondly, you know whether you have outliers or not. These are small. Usually, when it's small samples, you know each point. Okay, you know each point individually, and so you can easily, if there are outliers, first of all, you have to figure out why they are, and if they are really there, then you have the possibility of deciding that you want to summarize the data with the median rather than the mean, and you can make the informed choice. But unless you have good reason to use the mean. With very large samples, the inefficiency of the median even under the Gaussian assumption is much less important because your estimates, the means are going to be very close to zero. So the fact that they are 14% away on average doesn't make much difference. Okay. On the other hand, you will almost certainly have outliers. Okay. When you have large data sets tend to be dirty. Okay. So there will be points that are outliers. Okay. So if you don't look at the data and you just have to summarize them fast by single numbers, medians and MAD are better. Okay. So, yes. No.
But the outliers are like a small fraction.
This depends on the outliers. If you don't know, use the median. Okay. If you know, then fine, so you can make the decision. But if you want if you have millions of data points like you're going to have here, and you just want to have a number that would be the typical value, and you have the choice between mean and median, use the median. Okay, because you don't know what the outliers are. So by using the median, you defend yourself against outliers, and since there are so many data points, you don't suffer from the inefficiency. Yes?
How do I estimate the heaviness of the tail? How do I quantify?
So that's the point, okay? Numerically, you use kurtosis, but a measure that's called kurtosis, it's related to the expected value of the force power. It doesn't matter. The point is that for large samples, you're almost certain to have heavy tails. Okay, now for what does it mean about spikes? Okay, that's spike sorting. Okay, so we want to automatically threshold spikes. We are going to have millions of samples. Okay, just to give you example which is not atypical, we record now every day a few hours worth of data from 32 electrodes at 24 kilohertz or 25 kilohertz per second. So we have gigabytes of stuff. Okay, that's and then you don't want to do this by eye. Okay, you want to have measure. You want to do the spike candidate detection completely automatically. Okay, and then it makes sense to use medians and mods, okay, rather than means and standard deviations. Okay, the one thing to remember is that again, it's not exact, but the approximately 5 times MADS is 3 standard deviations. Okay, so you will see if large thing for Gaussian stuff is 3 standard deviation, okay, large things using the MAD would be 5 times the MAD. Okay, it's not exactly 5, it's 5 point something, but it doesn't matter. Okay, so 5 times MAD is 3 standard deviation. This is why you're going to see 5 MADs many times here. Okay. So I'm going to subtract the median from the memory potential to make sure, from the electrode signal to make sure that the median is 0. And here I'm selecting MAD, which is 5 times the median of RCV. By the way, the function MAD in MATLAB doesn't calculate the MAD. Okay, it calculates something else. I don't know why they did it, but it calculates something else. So I'm explicitly calculating the MAD here, median of the absolute value. Then I'm using find peaks like yesterday. Okay, and I want to have a look here at the spikes that got detected. Okay, so I'm drawing them. I'm drawing part of the signal. It's a very long stretch of signal, so you will see we will work at the end with order 10,000 spikes. So I'm looking only on a small stretch of it. So obviously I detected the large spikes. Okay, that's fine. Okay. Okay, here I'm not sure I wanted to detect that, okay, but we might be able to get rid of it later. So I detected this and that, and this, but I didn't detect that. Okay, again, okay, I detected this one, but I didn't detect that one. Okay, so obviously I didn't detect all spikes. Okay, this 3 standard deviation here might have been too conservative in order to find all spikes in the data. Now, there's a choice to be made here. Okay, you might say, and this is what we'll do at the end, you might say, I'm interested really only in the big spikes. Okay, and then that's okay. Or you might say, I want all spikes. and that's okay too, but you have to be then, there's a price to pay then. So next thing that I'm going to do is to try to look for the spikes, but using a less conservative criterion. So 2 times MADS, that would be what? If 5 MADS is 3 standard deviation, then 2 MADS is 3. Yeah, something like that. 6 over 5, yeah, 1.2. Okay, that's not a lot. Okay, and if I do this, so obviously I have many more spike candidates, much more red. Okay, and now, for example, here I detected these spikes. Some of them are the small stuff that I didn't want, but here I detected stuff that's obviously not spikes. This is obviously spike, I mean, look at it. This is a spike and that's not. What do you mean? Look, you see it. Okay, now that one I'm not sure about. Okay?
How do you know for it?
I am not sure. I told you I'm not sure.
How do you set the ground truth?
There's no ground truth. There's experience. Okay. But these are extracellular recordings. Okay, so I know I record from the neural tissue. Okay, I know there are big spikes, so presumably neurons are firing. Okay, so I have a lot of circumstantial evidence that the signals come from neurons. But with a single electrode in this type of experiment, of course, you can't know. Okay, nevertheless, you get a feeling to what the signal looks like when the tissue is dead. Okay, and it would be much flatter. It would be, there won't be this type of fast large events and so on and so forth. So there will be noise. Okay. Now, some of this noise is brain, and some of this noise is not. Okay, that's some of this noise will be machine noise. We saw this previously. I mean, you need to, but you know, okay, that after you did these experiments. So, story number 1, okay, when I first learned about extracellular recordings, I was finishing undergraduate degree in physics, and Elon told me about these things, and I was horrified. I mean, how can you make the deduction, okay, that these things come from a single neuron? And then Elon told me, eventually I got convinced, but the way that Elon expressed it is that there's enough circumstantial evidence to hang your grandmother. Okay, so לתלות את סבתא שלך. So that's there's a lot of circumstantial evidence that these are spikes, and eventually you get to you get to figure this out. What was the other story I wanted to say? I don't remember, maybe later.
There are data sets, maybe someone doing an intracellular recording and putting an extracellular recording near
Yeah, so the problem is that there are recordings like that. The problem is that simultaneously getting the spike intracellularly and extracellularly is very, very, very rare. Mickey did some recordings like that, you can ask him. Okay, the paper they published a few years ago during his postdoc included the simultaneous intracellular and extracellular recording with a multi-contact electrode. Okay, so you can ask him whether he has data like that. It's extremely difficult simply because the extracellular, you can't move the extracellular electrode while you have an electrode intracellularly. Okay, so usually you will put them a little bit with some distance between them so that they don't interfere with each other. Okay, and then you don't necessarily record from the same neurons. Anyway, that's presumably not spike. Nevertheless, it was detected. Okay, so what can I do in order to improve the detection of the spike candidates? High pass filter, that's right. Why? Not you, Omri. Why should? Yeah, so there are fluctuations that are slower than the spikes. Okay, the spikes are riding on top of fluctuations, and fluctuations make thresholding extremely tricky. Okay, so again, there's a trade-off here. Either if you want only the large spikes, then these fluctuations don't matter, okay, because they are small relative to the size of the large spikes. Okay, but if you want all spikes, okay, the large spikes are much larger than the fluctuations. Okay, so you can detect them easily, even in the presence of the fluctuations. But if you want the small spikes, then you want to get rid of the fluctuations. Very often you will want the small spikes, at least not in order to sort them, that's impossible, but in order to get a sense for the population firing rate around the electrode, okay, the multi-unit rate. Okay, so we want to get rid of the baseline. So that's obviously, okay, when we do spike sorting, we don't look at the data, get in trouble for finding the spike candidates and then high-pass filter. Okay, we always high-pass filter. Okay, that's something that we always do. I wanted to show you to do it this way in order to illustrate the problem. You must do the thresholding after your baseline is steady. Okay. So I'm going to, since we did this type of exercise a number of times, I'm just going to do it fast. I am calculating the power spectrum here. Okay, I'm using p-wells to calculate the power spectrum in order to see which frequencies contribute to the... So, these are the frequencies that causes these fluctuations. Okay, so first of all, you see 50 hertz, 100 hertz, 150 hertz, 200 hertz. Okay, these are the harmonics of the line interference. But the whole thing rides on top of something at low frequencies. Okay, that's a leftover of the local field potential. And standard number for filtering slow fluctuations when you want to do spike sorting is 200 hertz. Okay, rule of thumb. So I design a filter. I'm using FIR 1. My lowest frequency of interest is 200, and the sampling frequency is 20 kilohertz. So the period is 100. Okay, I should have put here 300 to, okay, filter of length 300. Okay, I'm going to look at its frequency response. Okay, so it suppresses enough at the low frequency end. Okay, I put also a high pass at 8 kilohertz because we know that above 8 kilohertz it's not biology. Okay, I didn't show you that it doesn't going to do much. Okay, but good practice is to use a bandpass filter when you're preparing yourself to do spike sorting, and then I'm filtering. Okay, so the one thing that I want, I didn't run one of the lines here. Okay, the one thing that I want to check is that the spikes roughly kept their shape. Okay, so it looks okay. The red spike is not smaller than the blue spike. Okay, there is a shift in the baseline that is expressed also a shift at the peak. It looks fine. Okay, so I'm using again my 3 standard deviation equivalent criterion. I'm selecting again the spikes, and we'll go out for we'll go for the break and continue now. Going to continue afterwards.
o sea, on je stan včera. Ne, jaz sprašujem, da je bilo to isto. Sprašujem, kako smo takimi, dosta takimi. On не, он я, конечно, мне кажется, что он сказал. Я вчера пошел к нему, потому что он меня это. И он сказал, что он еще машину долго как он из бесплатной машины. Он мне не знал, что она как слабая, как он говорит, нет, не слабая, покупать машину нельзя, потому что я ценности. Когда разница между всего там 3 тысячи больше, покупать машину себе, скорее всего, лучше. Да, лучше. Это тоже большая.
אוקיי. נמשיך? Let's continue. So, we got our spikes, and in a way you can see that the selection of the spike candidates is sort of more, in a way, more uniform. I'm still missing spikes, okay? These things are probably spikes, but they didn't get detected. Overall, it looks like I did detect everything, or most of the stuff that I call spikes. So that's, this is fine. Now, the problem at this point, okay, is that I'm going to have too many spikes. Just to show you, okay, I got 18,000 spikes, okay, and for the purpose of the continuation of this exercise, this is simply too much. So you might want them, but we are going, after we did all the efforts to get all spikes, okay, I'm going now to use a stricter criterion. Yeah, as I said, often you want to know things like firing rate of the population around the electrode, okay? And then you will want to have every single spike that occurred near the electrode. Okay, so it may be, this may be an important number, okay? If for doing spike sorting, okay, as a rule, you will anyway be able to sort only the large spikes. Okay, so in a way, there's no point in detecting all the small stuff because you're going to get rid of it at the next stage. So you may allow yourself to use worse, more conservative criteria. If you want a multi-unit activity, then this type of a bit more permissive criteria may be useful. Just make sure that you really have spikes. Okay, that's so I plot just to illustrate what's going on here. I plot my threshold. Okay, so this is the 3 standard deviation lines, the 5 mad lines. Okay, and okay, they detect a lot of spikes that are, you see, these guys are barely detected, and I know I will reject them at the next stage. Okay, so I'm going to look again for the spike candidates with a slightly more conservative criteria. Now, if you use this type of 3 standard deviation criterion, small shifts with your criterion are going to result in a lot of spikes. okay, because most spikes are small. Okay, so I shifted from 3 standard deviation to 3 standard deviation times 1.4. Okay, 4 point something standard deviation. But I could also, I mean, in our program, for example, for at least until the last generation of students, we used 7 MADs as a criterion for detecting multi-unit activity. Okay, but sometimes we use even 9. Okay, it's a bit a question of experience. Experience with the data and experience with what you want to do. So on purpose, I'm missing spikes here. Okay, but I do get the large ones. Okay, I'll get back to 7 because that's the number I've been using when I was preparing the example. Okay, now I'm going to extract the spike candidates out of the continuous waveform. Okay, and note, so I decide that I'm going to use, in this case, 64 sampling point. The sampling rate is 20 kilohertz, so 64 means a bit more than 3 milliseconds. That's quite a lot, and you will see it in a moment. Okay, it's important. In this case, there's a reason to it. So that's why I selected a relatively long interval. Usually 2 milliseconds, 1.5 milliseconds is good enough because these extracellularly recorded spikes are less than 1 millisecond wide. Okay, so we don't need huge amount of data. Another subtle point here is that I'm extracting them not from the filtered trace, but from the original unfiltered trace. And the reason here again is in order to keep as much as possible the original shape without affecting it by the filtering. Okay, so in this case again, there is an importance to it. You will see in a moment. Okay, so I extract my spike candidates. And I'm going to plot all of them. There are still a lot of them. I think even with the more conservative threshold, there are about 11,000 of them. So my computer is doing it a bit slowly. Okay, so here they are. What can you say about all of the spikes? Funny, unusual stuff that you see here. So first of all, you see sort of the autocorrelation of at least of the large spikes. Okay, so the large spike occur here and also occur at these positions. Okay, so you see the absolute refractory period, which is very short here. Okay, that's less than a millisecond. Okay, and you can sort of see the autocorrelation structure. Okay, what else? There's something that will become more obvious later, but the question is whether you can see it already here.
Very low peak. Bottom.
The what?
The low peak bottom.
This. Yeah, so we'll have a look at it. Yeah, so this will become, this structure will become more obvious in a moment. In a way, you can see it here too. Okay, you see this tract of spikes. Okay, we'll see them in a moment. Okay, this we discussed. Okay, so now we want to sort the spikes. Okay, there are many ways. The best way to sort spikes at the end is eyeballing, and I will sort of show to you here that now eyeballing what exactly? So there are, sometimes you eyeball the spike shapes themselves. Okay, so there are programs for spike sorting that provide you with a user interface for simply selecting groups of spikes directly and extracting them from all the spikes, and then you can slowly peel the well-separated spikes. Usually, we will not do that. Rather, we will use features. Okay, well, features are again things that describe the spike shape. Okay, that we'll talk about them in a moment, and then we do the sorting in feature space. Okay, and to do the sorting itself, we can build a tree of criteria. So first you separate spikes by one feature. Okay, you look at this at one of your classes, and then you separate it again, and so on and so forth. Okay, this can be done naively or not, or we can cluster directly in high-dimensional spaces using multiple features at a time. Okay, and finally, we can use a formal clustering algorithm. Give them the spikes or the features of the spikes and ask them to find the clusters. Okay, clustering algorithms work remarkably badly in spike sorting. Okay, they really do bad job. And I will run here only the simplest thing of the k-means, which is not very good anyway, but even under conditions in which you will see here, the clusters here are not bad for k-means. Okay, nevertheless, k-means will do pretty lousy job. Okay, so just what is a feature? Okay, feature is a function of the data. Okay, so you put a spike shape into a machine and you get a number. That's a feature. Okay, any machine works. Okay, so for example, a feature may be the voltage value at the peak of the spike. That's a feature. Okay, a feature is the width of the spike. A feature may be the value of the projection on the first principal component. That's also a feature. Okay, so you have a lot of different features. Okay, and we will try to use a number of them, a number of different ones. It's the third hour. Okay, we'll go through attempting to use a number of different ones. Okay, yes.
How do you assess how good they are?
We'll see. I mean, there are two ways. One of them is to get the histogram or the scatter plot and see that they define features. You have good clusters. Okay, that's the eyeballing approach. And I'll give you one way of trying to automatically determine whether a feature is good or not based on its distance from Gaussian distribution. Okay, so good features are features that are not Gaussian. Okay, so we'll do that. We'll look at that. Okay, but the question of feature selection is extremely hard. Okay, that's extremely hard means usually that there are no best general solution for everything.
If you had the ground truth, you could build it.
No, because the problem for a specific ground truth, okay, you can do supervised learning and get a best feature for that case. The question becomes whether these features are generally true for in other cases. In another experiment, for another electrode, and so on and so forth. Okay, and the problem of spike sorting is precisely that. Okay, you can find good features for any specific case. Okay, but it's difficult to find general rules that will give you good features. In one case, it may be the peak Memel potential. In another case, it would be the width. In the third case, it would be a projection on principal components. And the problem is that when you use all of them together, you start to get the clustering. You start to get noise. Okay, noise in the clustering. Clustering programs are not good at finding the informative features. Okay, they use the feature and they do whatever they do. If you work in a very high dimensional space with a lot of features, okay, then the geometry becomes extremely sparse and the program will do whatever it does. Okay, it will find something, which usually doesn't make sense. Okay. So let's eyeball. Okay. Obviously, there is a variability here in spike sizes. Okay. So the first feature that I'm going to look at is simply the moment potential at the peak, at the height, at the peak of the spike. Okay? Why? Because this is the point where I have a large variability in the data. And remember, variability is a thing for spike sorting. We want to find something that would discriminate between the classes. So I want to use points of high variability. Okay? So to see the classes, the tool again is a histogram or scatter plots. So here I'm using a single point, so I'm looking at the histogram. Wonderful. Okay? So the histogram immediately characterizes three peaks, three collection of points. Okay, there are the large spikes. Okay, there are middle-sized spikes, and there's this large collection of small stuff. Okay, so let's look at the large spikes. Okay, so I'm constructing a naive tree, a naive classification tree. Okay, so where do I want to put my threshold for the deciding where large is large? Okay, give me a number. 1.3. 1.25. Something, okay? We need to do something. We don't quite know, okay? I think I use 1.1. Okay, so I used 1.1. Okay, I wanted to have all the large spikes and then look at them and see. At this stage, I don't want to miss. Okay, so I'm using. I'm selecting the large candidates are all the spike candidates whose memory potential at the peak is larger than 1.1. Okay, and I replot them. Okay. Since this is not at this moment, I have some freedom in the construction of the tree. I'm allowing myself to be less conservative. Okay. So what can we say about this? These spikes that have a large memory potential, larger than 1.1. Okay, they seem to fall into two different classes. Okay, this is what you saw before. You remarked before. Okay, there are these spikes that have this negative stuff. Okay, the one that we saw before when we plotted all spikes were these guys, okay, that got detected, okay, and then this event occurred here. Okay, but if I just select the large spike, I see that actually this type of negative events follow large spikes. Okay, so I'm going to use now, I'm going to take all of these spikes and I'm going to divide them according to the values by a memory potential value around here. So I use point number 40. Okay, I want to look now at the histogram of the values here and use that to select a good point, which obviously will fall around that voltage. Okay, so I'm looking now at the histogram of the memory potential at point number 40 here among all large candidates. Okay, and again, there are those where the memory potential is negative and those in which it is not. Okay, so this make me classify the spikes into two classes. One of them are large at point number 32 and negative at point 40, and the other ones are the ones that are not negative. Okay, Okay, and here I sorted my spikes. Okay, these are the ones with the okay, so I have my two classes, these guys and these guys. Okay, now remember there are a few problems here that we will address in a moment, but I want to say one word about the physiology. This is cerebellum. Okay, and the large spikes all come from the same Purkinje cell. Okay, these are simple spikes and these are complex spikes. Okay, and the problem that the student who did this project, Liat, the problem she had to solve was to identify. So she was bringing the electrode so that she gets mostly spikes from one unit. So all the large spikes are presumably from a single unit. And then she had to decide which spikes are simple spikes and which are complex spikes. Okay, that's the problem. She brought this data to the course when I gave it a few years ago, and here is the solution. Okay, it's relatively easy. We could do it simply by hand. Okay, and we have the answer here. Okay, now there are some subtle points. For example, I still don't know what to do with the spikes here, which have the lowest Memon potential. Okay, I don't know whether they really belong to that class or not. They have some stuff here. Okay, it may or may not be important. Out of many spikes, there are maybe 5 or 6 that are in question. Okay, so this may be, for some purposes, this may be much better than you really need, and for other purposes, this may be bad, and you may want to identify these spikes as well. The point is that very easily by hand, I got these two classes. Okay, it's not a big problem in this case, just identifying these two classes. Yes, Alan?
Two questions. So first, on the lower graph, you can see that spikes are occurring pretty close to just after the main spike.
These?
Yeah.
Yeah, but still, you have here something that would be of the typical, that's about about half a millisecond, which is fine for the refractory period.
And small bumps before and not spikes?
There are spikes presumably of a different unit. Okay, they are much smaller. Okay, if you remember, there was an intermediate peak. This intermediate peak will give rise to these guys. Okay, they fire at as you can see here, at a higher firing rate than this neuron. Okay, and we'll see that we can sort them too. Okay, we can't. I don't think it's possible to identify them as simple or complex, but they do form a separate class. In a moment, you will see it.
And then you said that these are presumably spikes from the same unit.
Yeah.
But aren't complex spikes supposed to have bigger amplitude than simple spikes?
Intracellularly, yes, but here you see the extracellular expression. These are the sodium spikes that come. You can see extracellularly.
So you don't see the calcium towards the end?
No, you can't see the calcium spike. Roughly speaking, okay, the extracellular voltage that you record looks like the derivative of the intracellular membrane potential. Calcium spikes are slow. Okay, they're long and slow, and so they are expressed with relatively small currents extracellularly. The sodium spikes are fast, so you see them. If you remember, intracellularly, a sodium spike is about 1 millisecond long. Extracellularly, it's shorter. Okay, it's shorter because you basically see the rising phase. Okay, you see the derivative. Okay, now, even in this by-hand approach, okay, I can do better. Okay, I can do better because I did the selection here. I did a sorting by specifying a criterion separately on the peak and separately on this period of in which I identify the complex spike shape. But even here you can see, even in this plot, I linked the axis, so the y-axis is the same here and here. You can see that the complex spikes tend to be actually the smaller ones from this population. And again, this comes with the physiology. They ride on top of the calcium spike, so there's already some depolarization and some inactivation of the sodium channels. Okay, this is why they tend to be a bit smaller. So there is a correlation between the size of the peak here and the fact that this is a complex spike, and it may be better since I identified now two features. Okay, the voltage at that time point and at that time point. Okay, it may be better to actually cluster into this space, still by hand, but into this space. So to do that, I simply plot the scatter plot of the voltage values at these 2 points. Okay, so on the x-axis I have point number 32, that's the peak. So everything that's larger than 1.1, these were my large spikes. Okay, so these are the complex spikes and these are the simple spikes. Okay, because these have the negative values at point number 40. And you can see that these are a bit smaller than these. Okay, the peaks are here. And you can see that actually, instead of what I did, the way that I selected them was to put a straight line here, okay, and then to put a line here. Okay, you can see that when I did that, I actually put in some noise from here. Okay, so it may be better to put my boundary for selections as shapes in this 2D space. I can delineate better the group of the complex spikes and the group of the simple spikes. Okay, so let's do it. So I'm using here some tools of MATLAB, okay, that allow me to interactively draw polygons, and that's the IM-Poly. Okay, and it allows me to test, given a polygon, whether points are inside or outside. Okay, so these are MATLAB routines. Okay, we could, if you want to do a basic computational geometry, okay, you can try to figure out how to write a function like in polygon. It's not complicated, but so first of all, I'm going to draw the polygon. Okay, so let's start with the complex spikes. Okay, so I got the polygon. Now, there are kind of decision to make. For example, are these points among the complex spikes or not? Okay, I don't know. Okay, if I'm doing it interactively, I can later plot a polygon around them, look at their shapes, and decide what they are. Okay, things like that. Okay, with a good interactive tool, you can click on a point and it will pop out the spike shape. Okay, here I'm doing things very simply, so I leave them out. Okay, I want to look only at these spikes. Okay, so next I'm calling this function that provides the coordinates of the points of the polygon. Okay, and then I'm using here in polygon. Okay, in polygon you give a set of points, okay, x and y of a set of points, and you provide the polygon. Okay, the polygon is pos1, the x and y of the points of the polygon are in the array pos1. Okay, the x and y of the points I want to test, okay, are here. Okay, and in polygon, give me an array that has 0 and 1. 1 if the point is inside the polygon and 0 otherwise. So this line, Okay, simply extract the indices of all the points, and I'm going to plot all the resulting spikes. Okay, so these are the complex spikes. Maybe not all, maybe the ones on the side that I didn't select are also complex spikes, I don't know. Okay, but these are obviously, that's a good selection of complex spikes. Okay, let's select now the simple spikes. So I'm going to לדרוך על, to step on everything that I did. I'm not going to keep them separately. Okay, so and you can make different decisions here. I don't know what to do with the points that are on the edges. These points could be part of the polygon or not, of the simple spikes or not. These could be part of the simple spikes or not. If this is important to you, okay, you can test them. Okay, you can, with the interactive tools, you can test them. Let's get their position and plot them. So here are the simple spikes. Okay, all the simple spikes. And I can also look at other classes of spikes that are apparent here. So among the smaller spikes, okay, it looks like there's a separate class of spikes here. Okay, these are the spikes that made the peak of intermediate-sized spikes earlier. Okay, so you can see that on these, I can actually identify this class pretty nicely. I mean, there's a region of lower density here that allows me to sort of separate between it and the mass of small stuff. Okay, so I can try to identify these guys too. And again, you can make different decisions here. I'm not claiming that what I'm doing is the best. Okay, let's get them and plot them. So these are the intermediate-sized spikes. Okay, there's an interesting, a byproduct of doing this is an interesting relationship between the large spikes and these spikes. Okay, can you see it? Yeah, I mean, they tend, just before that spike, there's a tendency for these guys to occur. Then there's kind of a maybe lower probability, and again, a tendency of these guys. So there is some interaction between these two. Okay, to the extent, okay, that it may even be the case that these spikes somehow belong to the same neuron, although I don't think so. Okay, but it looks like there is an interaction. If they come from two different neurons, okay, there is some interaction between them. Now, what you see here is roughly what we call a cross correlation. Okay, a cross correlation, the firing rate of the large spike around times of occurrence of the small spike. Okay, so you see that this firing rate is slow here, it becomes higher here, then it drops to 0, okay, and then it goes up again. Okay, so there is a non-trivial cross correlation between this middle-sized spike and the large spike. Yes? That's true, but then why do you have high rate here and not here? Okay, here around the time of the middle spike, yes. Within this time interval, you don't know much. because you could miss spikes that overlap, with overlap. But you see this interaction at distances, time distances that are longer than the width of the spike. Okay, so it's not only an artifact of the spike selection. Okay, and the last thing that we can see here, let's look at these guys. Okay, they are large on the second, large and positive on the second time point. So I want to see who they are. Okay, so they are precisely these middle-sized spikes followed by a large spike. Okay, so those cases in which we have this type of interesting time relationship between the two. Okay.
It's both complex spikes.
Yeah, I mean the big spike can be either simple spike or complex spike following this. Yeah, I don't know. Okay, again, there's some kind of interaction between these two spikes that's non-trivial, and I can't give you any direct interpretation. But in a way, this is if you wish, the power of the extracellular recording. You see multiple spikes, you can separate them by shape, and then you can look at the interactions, and you know that these two neurons, if most presumably these are two neurons, okay, they are very close to each other. So you can see here a kind of, you can extract something about local interactions within the tissue. Okay, maybe one of them is one of the inhibitory neurons. I don't know. This is all okay, so all of that was done by hand. Okay, and at the end, doing it by hand is the best way of doing it. Always will give you the cleanest data. Again, if you don't want to do it by hand because there's a huge amount of data that you want to sort, then you want to use automatic classification technique. And here I'm sort of trying to demonstrate how to do it. So k-means is a MATLAB program that does k-mean sorting. Okay, you give it two things. You give it your vectors that you want to cluster. Okay, and you provide it with the number of clusters that you want the program to give you. It doesn't decide by itself how many clusters there are. Okay, just you tell it I want 6 clusters, and it will give you 6 clusters. Okay, I want it to cluster the scatter plot here. Okay, so I did it here by hand, and you can see that these clusters are well rounded. Okay, these are clusters that can be described by Gaussian bumps. I mean, everything like there shouldn't be, in principle, there shouldn't be a problem of finding these clusters. It's not a difficult clustering problem. So the way that k-means work is that it starts with centers, 6 centers that it selects randomly. Since it selects them randomly, every time you're going to run k-means, you're going to get a different answer, or potentially a different answer. Okay, so it throws 6 random points, and then recursively, iteratively does the following. It takes each point, okay, and it takes each point from the scatter and assigns it to the closest center. Okay, so once it does that, okay, you have 6. You can conceptually think of coloring each of these dots by different color, 1 of 6 colors. It takes all the dots that went to the same center and calculates their center. Okay, and these are the new centers. Okay, and then it does the same thing again and again and again and again until points don't shift between centers. Okay, when it has a stable classification, it returns. So, here it is. Okay, and what I'm going to do is to plot each one, spikes that belong to each one of the clusters separately in different colors. Okay, and then I'm going to also, I'm using here, gplot matrix is, you will see, it's a MATLAB routine for plotting multidimensional data. Okay, so that's the first class, second class, third class, fourth, 5, 6. Okay, and now it's going to plot here this is what gplot matrix is going to do. In a moment it will appear here. Again, there's huge number of spikes. So this time, so you can see that it found, so this is a scatter plot of our two variables against each other. Okay, the memory potential at time 32 and at time 40. Okay, and you can see that it found the two big clusters. Okay, here for the middle-sized cluster it failed. Okay, it did not identify it well. It's mixed up with the rest, and there is some sort of artificial separation of this of that one. Okay, now what gplot matrix does is you see here the histogram of variable number 1 by class. This is the histogram of variable number 2 by class. Okay, and these are the scatter plots. In this case, they are identical. Okay, this is 1 by 2 and this is 2 by 1. If you have more variables here, okay, if you ask it to have more variables here, you get the full matrix of scatter plots, of all pairwise scatter plots.
Where's the yellow cluster?
The yellow is these big spikes. Okay, the one that were small spikes followed by a big one.
What happens if we, could you run it with 5 or 4 k-means?
Yeah.
I mean,
Let's try. I mean, one of the nice things about MATLAB is that it's easy to run, and even my computer. Okay, in a moment, you see here the yellow ones are these black. That's the black class here. Okay, so again, class number 1. Class number 2. Class number 3. So now you can see that it failed to separate the, in a moment, yeah, that's a scatter plot. So what it did was to mix these two spikes together. Okay, and then again to do some sort of random separation into classes here. Okay, it's not for nothing that I asked it to have 6 classes. Okay, I ran this code many times already. As a rule, k-means is going to over-clump. Okay, so if you want it to separate your classes as relatively small classes, you want to ask it to oversplit. Okay, and then it will find your classes and also oversplit the large mass. And later you can try to correct. Okay, this happens often with clustering programs. Okay, you actually want them to overcluster, okay, and then by hand fix them. Okay, that's okay. So okay, let's try to overcluster by a lot and then go out for the break. So in a moment it will appear. Now we have 8 classes. So this is the first class, second class, third class, 4, 5, 6, Index exceeds matrix dimension. What happened? I don't know. It ran out of colors. Okay, let's try again. Okay. 1, 2, 3, 4, 5, 6, 7, 8. Okay. So it oversplit. Okay, it oversplit that one. But in principle, it's easy. These type of things are very often easy to correct. What it never does correctly is that one. Okay, the distance from this to the rest is too small relative to the distance from this to that. Okay, so it identifies all of these things every time. It identifies this as a single stuff, and it just puts arbitrary lines through it. Okay, according to the initialization. It did detect these guys, okay, tend to detect them. Okay, so, but of course, K-means is a very simple clustering algorithm. There are many clustering algorithms that are much better than that. Okay, the natural things to do here is to use a model that's based on a mixture of Gaussians. Okay, to write the points here as arising from a probability distribution that's a weighted sum of Gaussian with different centers and different covariance matrices, and then use an algorithm like the EM algorithm in order to do the clustering. So this would be, you know what I'm talking about. Some of you know what I'm talking about. Okay. There are. You don't need to go very far. Let's say after those of you who didn't take the machine learning course. Okay. Once you take the machine learning course, you will figure out that there are many different techniques that can be used in order to solve this problem, and that are pretty good, much better than K-means. Okay, even these techniques don't do the clustering very well, and in this case, is relatively easy. So easy that sometimes even K-means can solve it. Okay, let's take a break, and then we'll go back to actually to the question of feature selection.
זה נראה דוגמה מעולה למישור גם שמשחקים בול לשתי אבל כן לא כן, אם אתה מורח את זה לקיר מה שקורה במספיק קצת, זה בגבול. לא, זה טוב.
space יווני שאתה...
בכל המעבדה,
מגדיר לאן לאן.
מה שעשיתי כאן זה בעצם לעשות את ה-feature space ואז להסתובב עם פוליגונים.
זה אומר שאם אני אנסה, או מה זה, אם מישהו יארגן דאטה סט מאוד גדול, שבו כל אחד עכשיו היה ב-API, סורטינג באזור א' במוח, אם עושה את הביטוי כזה ואלקטרונות כאלה, ומישהו אחר באזור אחר, ואזור אחר, ואזור אחר.
החלק השני של הבעיה, זה המקום שבו דברים נהיים ממש ממש קשים. זה העובדה שהרישום משתנה תחום של הזמן. הזמן האופייני של רישום, מספק תאים שזה מביא את האלקטרודות של כמה משנה את זה לקח לה בעצם תוכנית זה החלק הקנוני של התמונות. לדעתי קורטקס אודיטורי יכול להיות קשור מהבחינה הזאת, כי בהם יש פעילות מאוד מסוכנת לאודיטורי, וקורטקס ויזואלי זה פחות חמור, מכיוון שהפעילות שם נוטה להיות סינכרון הרבה פחות טוב. באזורים גבוהים, קורטקס פרונטלי, אזורים כאלה זה עובד טוב. קורטקס אודיטורי יש איתו קשיים נוספים. היו שם קטנים וצפופים. בחתול למשל, שחשבנו גם ב-A1 וגם באזור משני, באזור משני לא הייתה לנו שום בעיה. תמיד היו לנו ספייקים גדולים וטובים יותר. כשאתה מסתכל על האנטומיה פשוט, אתה רואה שבאזור של פחות צפוף, תאים יותר גדולים פחות צפוף. קורטקס פרימרי באזורים פרסומטיים, יש לה נטייה הזאת, להיות בשכבה 4 שלך. שם מאוד קורטקסים סקונדריים, יש לה נטייה להיות פחות צפופים, אבל ישר כל הבעיה עובדת. הבעיה צורה במובן שאין לה פתרון, יש לך כלים שהם אנשים יכולים להשתמש בהם.
ספציפית לעבור רקורדינג ספציפי, אפשר להשתמש רקורדינג אחר,
אם אתה רוצה להיות בטוח שהסוף יצא טוב, מודול לכל הבעיות האחרות שנכנסו סעיף,
אז מה עם רישום רקורדים? האם כמו לדורת הגבולים?
לפעמים אני יודע רישומים באזורים אחרים, אבל אפילו בקופים, הבעיה של הסורטינג, כל דור ודור של תלמידים במעבדות בהדסה, הסיפור שלהם לסורטינג. אני יכול לעקוב אחרי המאמרים שלנו במעבדת סגל. כשלמדתי יהושע, אני חזר בתור סגל, אז חלק מהעבודת הדוקטורט שלו הייתה לפתח טכניקות לכיבוד סורטינג. היה להם התערבבות בספייקים שיצרה קורלציות והיו לזהות לא נכונות. בדור שלפניי, גם בגל היה סיפור עם קורלציות גם כן. כל דור ודור יש. זה נורא נגיד. טוב, יאללה. בואו נמשיך. Okay, so I want to get back to the issue of features. Until this point, we used features that are actually time along the voltage value at specific times along the spike, but there are many more features that can be used. So for this part of the lecture, I want to show you two families of features that are used quite often, and to say a word about feature selection. Okay, none of that, as a rule, both feature extraction and feature selection are hard problems. Again, hard problems mean that there's no good way of doing it. Okay, it's like it doesn't mean that there are no tools of doing it. It means that there's no optimal way of doing it, and a lot of different ways, and all of them work sometimes and don't work in other cases. So first of all, in order to do the feature selection, just for simplicity, I'm shortening the period of time that I'm looking at. Okay, I want my features to be sensitive to the important features of the spike. Okay, so the important features of the spike, as far as I'm concerned, are this main positive lobe and this negative thing that comes afterwards. Okay, so I just keep a short segment, now a bit less than 1 millisecond long, okay, that has these important bits that I know from before are the ones that define my spike classes. So the first family of features that I want to discuss are the principal component, projections on the principal component, on the principal directions. Okay, so I'm doing it here. The way that I taught you yesterday. So I first subtract the mean shape, and then I'm using SVD to extract both the principal vectors and the weights on the principal vectors. So you are the principal vectors, my features are going to be the V. Okay, V are the projection on the principal vector of each one of the spike shapes. Very often, people will not do this step. Okay, and they will do the SVD directly on the spike shapes. As I said, if you do that, then almost certainly the first principal component you are going to get would be the average spike shape, which may or may not be part of the story, but in a way, it's up to you to decide the way that you want to do it. I think that in order to describe the variability in the family of spikes, it makes more sense to do it that way. But admittedly, that's not the way that most people are doing it. Okay, so I did the SVD. Okay, and now I'm going to plot, first of all, the weights, the average sizes of each one of the projections, this diagonal of the matrix F. Okay, I'm going to plot the first two singular vectors, principal vectors, together with the mean spike shape. Okay, and I'm going to plot the scatter plot of the first two projections. Okay, just a moment. So these are the weights. Since I have 18, my vectors are of length 18, I have 18 principal vectors. And you can see here, these two, the first two directions seems to be to have large projections. Okay, the rest sort of go down linearly, and so when you see this type of slow change, usually you say these are unimportant. Okay, we'll see in a moment that they are indeed unimportant. Okay, but I will show you what they are. At the moment, when you see this type of plot, you say these two are important. We'll use them in order to define the shape. Okay, and the rest are presumably don't participate in defining the shape. Okay, so what are they? So yellow is the average spike shape. Okay, this is the first principal component, and that's the second one. Okay, now one thing that you can see here is that we don't get this type of derivative. Okay, which means that the spikes were aligned well enough. That's why I didn't, again, I know this data, so this is why I didn't align the spikes. Okay, if you get at this stage, if you get something that looks like the derivative, then it makes sense to align the spikes and redo the analysis as part of the pre-processing. Here, neither the first nor the second principal component look like the derivative, so presumably the spikes were well aligned. The first principal component tell you something about the size of the spike, okay, and the second one, okay, seems to tell you, note that this is 0. Okay, the second principal component is all negative. So if you add a weighted sum of that to that, what you get is a decrease in spike size and also a decrease in the tail. Okay, so presumably large weights on this will define our class of large spikes, and in addition, large weight on that will define the complex spikes. Okay, this is basically the story. Okay, so let's look at the scatter plot. This is first principal component, second principal component. Okay, large here means always large in absolute value, because the sign, okay, the sign both of the principal vector and of the weight on the principal vector is not defined. Okay, it can be, you can multiply both by -1 and get exactly the same decomposition. So you can see that this and that, okay, both of them have large negative weights on the first principal component, and in addition, this has large negative weights on the second one. Okay, so presumably these are the complex spikes and these are the simple spikes out of the large spikes. Okay, that's we can do. I mean, I'm not going to do it, but actually I will do it. Okay, let's run k-means. Okay, same procedure as before. So first class, second class. So these are large spikes, the red ones. Okay, and I don't know why we don't see the yellow ones, but you can see in the scatter plot that it did as well as it did with the voltage values. Okay, so also the scatter plot, if you remember the scatter plot for if you remember the scatter plot for point number 32 versus point number 40 that we had in the previous hour, it looks pretty much the same as that one. Okay, so what is the advantage of using, and here you have this class of middle-sized spikes. So what is the advantage of using that relative to the voltage values? The advantage is that this is automatic. Okay, and not for nothing, it's not for nothing that the SVD extracted these two components. Okay, and we saw it by eye, and the SVD extracted the same thing. Okay, SVD is the advantage of using the SVD is that you don't need, again, you don't need to analyze in great detail the shapes that you get. It provides you with directions that are presumably good directions to start with. Okay, so the first two principal vectors define the same type of variability that we did by eye before. I promised you to tell you what are the small eigenvectors, the one that goes from after number 3. So here I plot all the eigenvectors as in color map. So that's the first one, okay, with the peak. Okay, that's the second one. Okay, and so this is 3, 4, 5, 6. What can you say about them? What do they look like? But noisy yeah, there is an increase in frequency here. Can you see? So for example, this one has 3 yellows, sort of 3 blue, 2 yellows, 3 blue, 4 yellows, 3 blue, 3 yellows. If I go here, it's 2, 1, 3, 1, 2, and so on. So that one seems to fluctuate faster than that one. Okay? So let's, for example, look at number 7 and 8, this one and that one. Okay, I'm plotting them now as a line graph. Okay, what can you say about them? They actually have slightly different frequency. You can see that these peaks are closer than these are closer than here. Okay, so the red line has slightly higher frequency than the blue one. Okay, so in order to get out the periodicity, I simply calculate the Fourier transform. Okay, and I'm going to plot the Fourier transform. So this is the Fourier transform of the first one, second one. This is the number of the eigenvector, and this is the frequency. I have only 9 frequencies because I have 18 data points. So you can see that the frequency tends to increase. Okay, as I go to higher and higher order eigenvectors, and they sort of come in pairs. Okay, why in pairs? The cosine, yeah, the cosine and sine version of the. Okay, so the high order eigenvectors, principal components, are actually sines and cosines to a first approximation. What does it mean? I mean, where do these sine and cosine come from? So, fact is that if you take a stationary process, okay, and you do a PCA to a stationary process, you get out exactly sines and cosines. Okay? Why? Again, for those who come from physics, the principal components are eigenvector of the covariance matrix, and the covariance matrix is Teplitz and constant on diagonal. Okay, and a covariance matrix that constant on the diagonal has eigenvectors which are sines and cosines. Okay, so it's not approximate. Okay, a stationary process will always give, if you apply principal component to a stationary process, you will get sines and cosines. So if you get a picture like this, okay, that your higher order principal components look like sines and cosines, okay, you can know that you extracted all the structure there is. Okay, so I know, okay, for this data set specifically, for this spike specifically, I know that all the information is in the first 2 eigenvectors and that there is no additional information. Once I subtract the contribution of the first 2 principal vectors in this case, okay, all I get is a stationary noise. Okay. Let me illustrate this. I'm going to generate a stationary process using techniques that we already know. Okay, so I'm generating a bandpass filter. Okay, and then at each, I am generating a lot of trials, okay, 1,000 trials of white noise that I filter through the filter, through the bandpass filter, and then I'm calculating the. Actually, I didn't use the window here. I'm calculating the power spectrum here. Okay, so this is the power spectrum of my tiles. Okay, and I know that they are stationary because this is the way I generated them. Okay, I just took stationary noise and put it through a filter. I removed the beginning. Okay, I removed the beginning because I wanted to get rid of edge effects. Okay, so I have here 1,000 samples of a stationary process. Okay, now I'm doing the SVD. Actually, I want to keep figure 3. So we'll put this here. So I'm doing the SVD. Just a moment. So first of all, this is the correlation matrix. Okay, very, very stationary. Okay, unsurprisingly, because that's the way that I generated the data. Okay, these are two of the eigenvectors. So they never come exactly as sines and cosines because, well, there is a finite size effect. Okay, if I would have not 1,000 but 1,000,000 tiles, then there would be more sines and cosines. But if you have eigenvectors that are highly fluctuating like this with a slow envelope, that's a good sign, good evidence that you get a stationary part of the data. Okay, these are the eigenvectors. Okay, the eigenvalues. So these are the weights of the size of the projection on the different eigenvectors. Okay, and now what did I have on figure 3? No, this I wanted to keep. So I want to open 2 new figures now. So in a moment, so these are all the eigenvectors. Okay. You can see that they are sort of sinusoidal here, but it's hard to follow the frequencies. In this region, it's very easy to see that there are actually sines and cosines. The frequency that increases, you can see it. Okay, this one has slower rates. Okay, and as I go in, they have faster and faster rates. And here something else happens. They start to get mixed up in funny ways. So again, I'm doing the Fourier transform of each one of them. And this is what's plotted here. So first of all, they tend to have a clear peak in frequency. Okay, so they are approximately sines and cosines. Okay, in this range, they tend to have either that frequency or that frequency. Okay, or sometimes they are a bit mixed. Okay, in this range, they have a clear single frequency, and then again at the high frequency, they tend to mix up a bit. Okay, why? Okay, this has to do with the power spectrum. Okay, basically what you see, the eigenvectors, the eigenvalues. Okay, this diagonal of S basically follows the power spectrum. Okay, so the higher the power spectrum, the larger the projection on that sine wave. Okay, so the first principal component will have a frequency that's approximately here at the peak of the power spectrum. Okay, the next one will get selected from this region, and then they will start to get selection from that region, and then from that region, and so on. Okay, so this is what you see here. Okay, the first eigenvector has a frequency that corresponds to the peak of the power spectrum. Okay, and then you start to select frequencies that are go towards 0 and toward higher frequencies. Okay. Next, we are in this range. Okay. In this range, the ordering of the frequencies is clear. Okay. You go up in frequency, you get lower and lower values of the power spectrum, lower and lower eigenvalues. Okay. So that's this range. And finally, once the power spectrum becomes very small, okay, there's again no way of selecting the right frequency. Okay, so the eigenvector starts to be sums of a few frequency components. Each one of them has a random combination of some frequency components. Okay, this corresponds in the power spectrum, this corresponds to this flat region. Okay, and this is what you see here in the time domain, and this is what you see here in the frequency domain. Okay, so they correspond to the flat region of the power spectrum. So, again, if I would have 1 million tiles, then this picture would be even clearer. Okay, I would get a single frequency component on each one of the eigenvectors. What is the lesson? Don't do PCA on a stationary process. You know the answer to start with. It's just sine waves ordered by the power spectrum. Okay, and it doesn't help you more than doing the Fourier transform. Now, you will find what? Because Fourier is much simpler. It's faster, simpler, and it has other properties that are okay. It's the answer, so why do the approximation if you wish? Yeah. Okay, so we got now at least two classes of features. One of them is are the voltage values. Okay, the other one are the projection on the principal components. Okay, the SVD values. And now we want to select. We need to select. We have a lot of them. Okay, I showed you in this specific case that I wanted to use only the first two principal components. But among the voltage values, for example, I would have liked to be able to select them not by eyeballing, but by some kind of principled method. Okay, so the idea here for feature selection that I want to, this is one example of an approach for feature selection. The idea is that features that you can use in order to do well the spike sorting are features that gives you a histogram that's strongly non-normal. Okay, why strongly non-normal? You want it to look like that. Okay, with a lot of peaks. In particular, it's not a single peak. It's not unimodal. Okay, so one way of getting features that have some chance of helping you to sort data is to look for the features that are as far as possible from Gaussian. Okay, so how do we find features that are far from Gaussian? Okay, the standard approach to do that is to run something, to calculate something that's called the Kolmogorov-Smirnov statistic. Okay, that's a complicated name for a concept that's relatively simple. Okay, so I take this histogram. Instead of working with a histogram, I'm going to work with the cumulative distribution. Okay, so I'm going to take the histogram and, if you wish, integrate it. Okay, get the cumulative distribution. Here's how to do it by hand, but simplest way is simply to use the MATLAB routine. ECDF is empirical cumulative distribution function. Okay, you give it a sample of values, and it gives you back the x and y values, okay, for getting the cumulative distribution. Okay, the sample that I provide here is not the histogram itself, but the histogram after I normalize it to have mean 0 and standard deviation of 1. Why? Because I'm going to compare it with a normal distribution with mean zero and standard deviation of 1. Okay, so first thing that I do is to normalize my distribution to have mean zero and standard deviation of 1, and then I calculate the cumulative distribution function and I plot it. Okay, so this is a cumulative distribution function. Okay, how do I see that it's strongly non-normal? Yeah, it's very far from, it doesn't really look sigmoid, okay, it has acceleration and decelerations, many of them, okay, one for each peak. Okay, so how do I compare it with the normal distribution? I'm going to plot on top of that, on top of this plot, I'm going to plot the cumulative distribution of a standard normal. So I'm using MATLAB. Okay, norm CDF gives you the cumulative distribution function for the normal distribution. I want it calculated at point X that I got here from the ECDF. Okay, and I want it to have mean zero and standard deviation of 1. So normal is my normal cumulative distribution function. And now I plot both of them on top of each other. Okay, so now what is the case that the Kolmogorov-Smirnov statistic is simply the largest distance between these two. Normalized appropriately, but normalization is unimportant here. Okay, so the larger the distance between the cumulative functions, the further away from normal my data is. So this is going to be my statistic. What? This is not a heuristic. This is the Kolmogorov-Smirnov statistic has a theory. Okay, a very good theory. It's a way of non-parametrically comparing two distributions. If you have two sets of numbers and you want to test whether they come from the same distribution or not, okay, that's a test that you can run. You calculate the empirical cumulative distribution of both and you look for the maximal difference. Okay, and if they come from the same distribution, then there's an exact non-closed form formula for the probability of the distribution of the distance, so you can create an exact statistical test out of it. It is used to some extent, not extensively, but to some extent. The idea here is not to do the test itself, not to calculate probabilities, but to use a statistic, which is the distance between the distribution, as a measure of the non-normality. Okay. There are other ways of calculating distance. That's not the only way to calculate distance between distributions. You can use things, again, for those of you who took עיבוד מידע ולמידה, you can use mutual information between the two KL divergence, I mean, between the two distributions and things like that. All of them are to a first order the same. Okay. This is a simple way of doing it. The problem with the information theoretical measures is that you need to calculate histograms and take ratios and log, and you have zeros and infinities and all kinds of things like that, so you have to deal with these issues. This one, the advantage of that one is that you don't need any binning. Okay, it's based on the empirical cumulative distribution function, so no binning at all. Yes?
So the input for Kolmogorov-Smirnov is always the CDF? You have to always...
No. It's not the CDF, it's the normalized distribution. It's your sample. Okay, in this case, in my case, okay, I normalized it to mean zero and standard deviation of 1. Okay, because I want to compare it with a normal distribution with mean zero and standard deviation of 1.
But it seems like if you wouldn't work on the space of the CDF, so the units would have a totally different meaning, right?
Which units?
If you will leave it as it is, or just normalize it without creating the...
No, instead of normalizing it, I could calculate the mean and standard deviation and ask for the normal distribution with that mean and standard deviation. And then I will work in the units of the original, of my original measures. Okay? And I would get exactly the same KS statistic. Okay, it would change the x-axis. It would shift and stretch the x-axis, but it would not modify the shape of these functions.
Why are you using CDF and not CDF?
Because for that you have for the CDF, the KS theory works for the distance between the CDFs. This is where it's simple. Okay, it's more complicated when you look at distances between the PDFs. Okay, it's just, I mean, we are using, again, there are many different ways of calculating distance between distributions. This is one of them, and it's a good one in the sense that it has a theory. The fact that it has a theory is in a way irrelevant to what we are going to do with it. Okay, we are just going to use this as a way of estimating the distance between distributions. Okay. If you wish, this distance is a mathematically defined distance. It has all the properties of distance functions. You can define a distance on the space of all distributions using this. I mean, it's a real distance, okay? But it's so in math and statistics, it's used quite extensively. Okay, all of that is irrelevant here. It's just the fact that it's a good way of estimating a distance, a good and a simple way of estimating a distance between distributions without making all kind of arbitrary choices like you have to do when you use probability distribution functions. For example, size of the bins. Okay, the size of the bins is going to affect your numbers. Here, the bins don't come into, there's no binning. Okay. Okay, so I'm not going to actually calculate these distances here. Instead, what I'm going to do is to use a MATLAB routine called KS test, Kolmogorov-Smirnov test, that by default does this comparison. So if you call KS test with your numbers, with your original numbers, okay, with your normalized numbers, it will give you the KS test for the distance between this family of numbers and the normal distribution. Okay, so it does the test, okay, it provides back the answer and the p-value, but they're irrelevant here. Okay, so I'm going to do this. First of all, for all the time domain points. Okay, all the 18 time domain points. So I'm going to calculate the K statistic for all of them. And this is the answer that I get. So this corresponds to the peak of the spike. Okay, it has the largest non-normality index, if you wish. And then I have, nearby points are almost as good in terms of non-normality. Okay, and then I have another set of points, the points which are further on in time. These are the points that discriminate between the simple and complex spikes. So they are again have a relatively high non-normal distribution. Okay, so now the question, I need to select features. Which features would be useful to select and which are given that graph? So one thing that you want from a feature is that it will be non-normally distributed. Okay, that's this rule of thumb that I introduced here. But if you want to use multiple features, okay, you also want them to be uncorrelated. Okay, so that they would provide independent information. Okay, so I did it here in a way that's a bit complex. So first of all, I sort all the non-normality distributions by order. So I here, this vector I, is going to contain the indices of the indices of non-normality by order. Okay, so this is 0.5 and then 0.6 and 4. Okay, and then it goes here and selects 17 and 16 and 18 and 14 and 15, so this region, and then all the rest. Okay, point number 2 has the least non-normality index. Okay, next, what I'm doing here is to plot the correlation matrix of all time points with each other, sorted by they are now sorted in this order. Okay, so they are sorted by the size of by the size of the non-normality index. So you can see that the first three, for example, although all three of them have relatively high non-normality index, they're also highly correlated. Okay, so there's no point in using more than one of these three, okay, in the sorting algorithm, because they provide the same answer. Okay, then the correlation drops to 0 later in time. So the next point that you might want to consider is point number 17. Okay, so these are the 2 points that point number 5 and 17 would be the 2 points that are, according to this, the best to use for the sorting. If you remember, before we use, so point number 5 here is point number 32 from before. Okay, so we use point number 5, and point number 40 would be 5 plus would be 13. Okay, so what this thing tells me is that actually it would have been better to use not point number 40 but point number 44. Okay, it would be a bit better. If you remember, the size of the non-normality index is pretty much the same for all of these points. Okay, so point number 13 is here, which is 12. Point number 13 is here. So actually, again, eyeballing, I selected the 2 best points, okay, that are also independent of each other, as given by these criteria. Yes?
Since they're not normally uncorrelated, really know
Uncorrelated doesn't mean that they are statistically independent, of course. Okay, but that's the simplest way of selecting, having a chance of them being independent. Okay, if they're independent, they are uncorrelated. Okay, so your first guess at being independent would be your first choice would be to take the ones that are uncorrelated, and then you can ask which one among the uncorrelated is the least independent. You can also do tests for independence, but they are much more complicated. Okay, that's what I was trying to show here is that these actually work in a way. Okay, so I'm not going to do these k-means again because that's exactly the same, pretty much exactly the same k-means that we did before. Unfortunately, the next section of the presentation was about yet another family of features which come from wavelet decompositions of the spikes, but time runs out and they're actually not as good as the ones that we got here. So I'm going to skip that. You can look at you have the presentation, you can look at it. These are, I was using here something called the Haar wavelet, H-A-A-R. The Haar wavelet, in order to decompose the spikes, you get a set of features, and they're actually less good than either the time points or the SVDs. So you can look at them and see how they look. To summarize, okay, for analyzing shapes, first of all, you need to pre-process the data correctly. Okay, so the first, we saw all kinds of things that we need to do. First of all, for detecting the shapes, you need to process the baselines. Everything here is on a case-by-case basis. But if you want to select features, your shapes by size, you need to make sure that your baseline is correct, and so on and so forth. Okay, we can analyze the variability between shapes. Okay, the properties of the ensemble of shapes using principal component analysis. The tool would be SVD. Okay, you have, once you start to do your SVD, you might want to do additional preprocessings of the type that we did yesterday, for example, aligning the shape correctly. If you don't align them correctly, you start to get these derivative-like components. So diagnostics for bad alignment is the occurrence of derivative-like components. And then for clustering, feature selection, I introduced this heuristics of maximal non-Gaussianity measured here by the KS statistics. All of that you probably feel is sort of a lot of rule of thumbs, okay? And that's in the nature of the problem. Okay, the problem is badly defined, okay, so I can only show you how it works in specific cases and suggest tools. When you come, what I showed you are the major tools. Okay, there's a lot of additional stuff that you may encounter as time goes by. If you master these techniques, then it will be easy for you to figure out what the more complicated stuff is doing. So this is it. Okay, tomorrow we meet for the last workshop. So what we will do, basically what we did today on the RET recordings, pre-processing, shape extraction, and then sorting by hand, by SVD, whatever, as much of it as you will succeed in doing in the 2 to 3 hours that we have, and that's it. That's almost the end of the course. Okay, so see you tomorrow.
אני יכול פשוט לקחת אותם איך שהם, בלי להעביר אותם ל-PDF, בלי כלום, את הדאטה, ורוטינה, כן, עובדת על המילים, זה הזמן וחושבת בתוך המקנים. אז המאגלב כבר הופך את זה ל-PDF. בעצם המכניזם הוא כזה שזה נהפך קודם ל-PDF, לא הפעולה